Okay, started the recording again. All right. So a couple of things before we get started here. Obviously, lots of new assignments to do. And we actually poorly communicated, I feel like, with each other because from my understanding, it was only going to be one added, and it ended up being three, two of which were labs, which I thought we agreed at our last GSI meeting we weren't going to do, but here we are. But in that case, I thought it would be a good idea for this one in the next lab to basically give like a mini lab lecture like I did before. So at least I can build on the conceptual understanding for these experiments a little bit. Because I know it's difficult sometimes to do one of these lab write-ups and this data analysis when you never actually did the lab physically yourself. So yeah, like I mentioned earlier to everybody, feel free to interrupt me at any point if you have questions or in chat will work as well if you don't have a mic or if you want to be a little more discreet about it. But obviously your questions, somebody else might have it as well. It'll probably help everyone to ask them. Okay, so Lab 8. It's on absorbance spectroscopy and fluorescence spectroscopy. One of these you've done already, which is absorbance spectroscopy. In a previous lab where you used UV-Vis. The other fluorescence spectroscopy very much related. It's an emission process, so it's very much related to the atomic emission lab that you did as well. Okay, so what's going on here? Well, I'm gonna kind of jump way back to the very basics. And we're gonna talk about how molecules store energy. So if we're just looking at these classically, like classical physics, there's many ways that molecules can store energy, but the four most common and easiest to understand, simplest models are the ones on the screen here. The first is translation, which is linear center of mass motion. So basically, a molecule's center of mass can move in the XYZ, and that's what we call translation. Similarly, we can store this energy through another type of kinetic energy called the rotation, which is where if you're just looking at this molecule, not the pink one, that's doing translation actually as it comes in. But if we're just looking at this molecule here, it's rotating. And this is a linear molecule, so there's really two rotational degrees of freedom, two axes where we can rotate this molecule kinetically to store energy. So that's another form. We also have vibrations. So anywhere where these things are bonded, the distance between the two nuclei, basically, like this is water, right? So the distance between these two nuclei fluctuates. We consider this like a spring between the two atoms. It does very much behave like a spring system, and we call these vibrations, right? So you can imagine vibrations become the number of vibrational degrees of freedom becomes very large as the number of atoms in the system increases. And then, of course, we're chemists, so we don't just care about the nuclei because these are all nuclear motions, right? We also care about what the electrons are doing. And we have basically energy that's stored through the electrons. So electron kinetic energy is usually not considered, even though that is a part of energy. If you consider the kinetic energy is proportional to the mass, and the mass of the electrons is basically negligible compared to the nuclei, we don't really think about the kinetic energy of the electrons very often, unless it's a free electron floating off on its own away from the nucleus. Instead, we consider the potential energy based on how close it is to the nucleus, because the electron's negatively charged, the protons are positively charged, right? So these are different ways that we store energy, and these are ones that you're going to keep coming back to. Now, in actuality, it's a lot more complicated than that. So we almost always have more than one of these ways of storing energy at the same time. Here you're seeing rotation and a vibration at the same time. Here you're seeing translation, vibration, rotation all happening at the same time, and interaction between multiple molecules. These are a bunch of water molecules coming to form hydrogen bonds. So if I back up real quick and then show it again, they start off and they're kind of coming together, and there's intermolecular forces acting. So these are dipole interactions, which is a different type of energy storage that I didn't even talk about before. And you have chemical reactions that can happen. This is actually a very neat chemical reaction in the field of chemical dynamics called a roaming mechanism, where this methyl group gets removed due to rotational energy imparted from this collision. It comes in, it imparts rotational energy. That gives this methyl enough rotational energy that it can actually dissociate from that atom and go to this one. So these are all, it gets really complicated really fast. And to make matters even worse, right, these are all classical models of what's going on, but we know that there's actually quantum mechanics involved as well. So all of these types of motion, and you guys are going to explore this a lot more when you take PChem in the future, are quantum in the way they work, because matter is essentially a wave. So it all follows the rules of quantum mechanics. And we have things that are small enough in mass that we actually see the quantum that's going on. And each one, for each one of these, there's a basic model. So for translations, it's particle in a box. For rotations, it's the rigid rotor. For vibrations, it's called the harmonic oscillator. And for electronic transitions, or electronic potential energy, that's the hydrogen atom. You're going to solve the Schrödinger equation in PChem, specifically your quantum section, for all of these all of these systems in class at some point, right? And each one is going to give you different energies. And so what you get out is, well, first of all, quantum means that there's only certain energy levels accessible. So if I go back and I'm thinking about this vibration that's going on here, right? It's only allowed to vibrate at certain frequencies and not in between others, right? So there's only certain energy levels allowed, right? That's a really weird thing when you think about it, especially when you're thinking about translation, right? If you think about some guy running, right? He's only allowed to go 1 mile per hour or 2 miles per hour, but he can't do anything in between. That's a really weird concept. But that's how things work, right? In a quantum world. So what's interesting is all of these transitions, right, have different energy level gaps in them, right? So if I'm ever doing an electronic transition, right, these gaps tend to be, depends on the molecule, but from going from one, like your ground state electronic orbital, your HOMO, your highest occupied molecular orbital, to your lowest unoccupied molecular orbital, HOMO to LUMO transition, right? These tend to be about 4,000 to 12,000 wave number. Chemists really like to use wave number as a unit of energy. If you're a P chemist, you probably use EV a little bit more, just because we're almost always talking about electronic, and we like to have nice numbers that are, single digits. But in general, wave number is probably the most common unit of energy that chemists use to describe these sort of things. But essentially, you have this 4,000 to 12,000 wave number gap in energy, right? And you get that from solving the Schrödinger equation of the hydrogen atom. Similarly, when you do the harmonic oscillator one, you see that there's vibrational levels inside of these electronic ground states, right? So I can actually have a transition that goes from the ground electronic state into a higher order vibrational level, like V equals 3 in an upper state of here. So there could be a vibrational level here. So I'm not limited just by this arrow. I could have an arrow going in between. Similarly, there's rotational levels inside of that, which have even smaller gaps, and translational energy levels, this is not even drawn to scale, are much, much, much narrower. And we find that they're so small, in fact, we usually just treat it like a continuum of energy. Now, the consequence of this is that by using different types of light to different parts of the electromagnetic spectrum, we can probe different types of motion in these atoms, different types of ways of storing energy. So if we want to look at the electronic transitions, we're typically using UV invisible light. That's about the energy regime for that. Infrared, similarly, will excite vibrational energy levels within the same electronic level. Microwaves will excite rotational inside of the vibrational. And then it's not very practical, but this energy is so small, you would need things that in the electromagnetic spectrum, if you look, wavelengths that we would call long waves. And we really don't have any practical way of doing that. The closest we have is the LIGO detector that detects gravitational waves. You would need something like that to be able to start measuring individual translational energy level transitions. So it's not really super practical to try and do these sorts of measurements. But in general, you could take a UV vis spectrum, and we do this all the time, and get vibrational and or rotational resolution from like a high energy laser by doing these sorts of things. There are, like I said, other ways of storing energy. One is what we're going to talk about next week. So I'll be adding to this slide basically next week when we talk about NMR, where we're talking about the spin flips of a nucleus. So spin flipping itself is, at least nuclear spin flipping, is in the megahertz range. It's radio waves that we use to actually detect, or to actually create a spin flip transition. And we'll talk about how we do that next week as well. So when we think about the different kinds of transitions, we use these things called Jablonski diagrams. They're all over in Harris, the textbook you use, specifically in the spectroscopy chapters. But in general, these are the most common types that are talking about going from, this would be S0. So this is a singlet ground electronic state. S1 would be your singlet excited electronic state. And then these are the vibrational levels inside of each one of those. Also, there is a triplet excited state where you have a spin flip happening. This is considered a forbidden transition. You'll talk a lot more about what that means in PChem. If you have any questions, you can ask me. But this tells you about all of the potential transitions that are possible in this system. So essentially, initially you have to have some sort of absorption process that puts you into one of these levels up here from S0, right? You can have a vibrational relaxation. So as we go, like these straight arrows pointing up, these are radiative transitions, the way this specific diagram is drawn. There's all kinds of versions of these, but these are the rules for ones that look like this. These are radiative processes. So a photon has to go in to do this. For fluorescence, which is one of the spectroscopies we'll be using, that's where a photon comes out in this transition. And if you notice, you see this vibrational relaxation. The curly arrows are non-radiative relaxation processes, essentially. So vibrational relaxation does not occur through releasing a photon. It happens through collisions of other molecules. Basically, a molecule gives some of its energy to another one, and then it can it can emit a photon and relax back down to the ground state, right? There's also another thing called intersystem crossing, where a spin flip can occur, and in general, triplet states are lower energy than singlet states. Not as low as the ground state, but a spin flip will occur. And again, this is through the loss of energies through sometimes collisions, but also there's other processes that can happen. You can get very, very complicated very fast. And then you can radiatively transition from the triplet down to the ground state, and this is called phosphorescence. So there's a bunch of different ways for this energy to go and to relax and to change into different energy levels. But what's important and a key here is that some transitions are more intense than others. And this all has to do with the probability of these transitions. This comes from something that's central to quantum called Fermi's golden rule. You won't see this until you take upper division quantum mechanics, either in physics or in PChem. You'll definitely see this here at Berkeley when you take PChem. They'll probably derive it in class, but won't ask you to derive it. If you go to grad school and take a graduate quantum course at any point, you will absolutely have to derive this. If you go to grad school for PChem, you will undoubtedly be asked this on your qualifying exam. Every physical chemist I know is asked about Fermi's golden rule and how you get there. This is a fancy way of hiding an integral, so there's a lot of calculus that goes into this. If you really want to know, I can play with this for you, but it's a little too advanced for this discussion. But the reason I'm showing it to you is this is how you get Beer's law, right? This probability of an MN transition is basically from going, let's say it's the absorption, it's the probability going from this energy level to this energy level, right? And each one of these transitions has its own probability. That's proportional to this matrix element, which has to do with the electric potential of the states. Okay, so how do we get Beer's law from that? Well, Fermi's golden rule really leads to Beer's law when we go macroscopic. So here's Fermi's golden rule, and it also is proportional to what I would say is 10 to the power of this epsilon, which is from Beer's law. It's called the molar absorptivity. Other sources call it the extinction coefficient. In your lab handout, they call it both back and forth constantly. So if you're like, what is the extinction coefficient? They call this the molar absorptivity before. They're the same thing. They're just different names for the same thing. But basically, it's this variable that depends on anything that has to do with the electric field of the energy levels you're talking about. So the temperature, the specific wavelength being used to do this excitation, the solvent molecules around it change the electric field potential, the size of the molecule itself, all of these things go into that. When we think about it, it ends up being 10 to the E, and this is just unit conversion stuff. But really, how this applies is, okay, the probability of a transition, right, is how much. If we go back real quick to the Jablonski diagram, and we see this is our photon energy, right? So this is the wavelength of energy that we're putting in. We're putting in a whole bunch of these photons, right? Some of them get absorbed, some of them don't. And the intensity has to do, or the intensity essentially is the probability. It's the ratio of the photons that got absorbed versus the total number we put in, right? Another thing we call that is the transmittance, this ratio of the intensity, right? The number of photons that we put in, I0, the number of photons that got absorbed in that time frame. We call that the transmission. It's exactly equivalent to this probability, right? Similarly, it's kind of nice to talk about this in terms of the log of T, because there's a scaling that has to do with the concentration that's first order kinetic, basically. So we can eliminate our exponential by taking the log of this transmittance. So it's usually better to talk about the absorbance, which is just defined as the log of this ratio, the log of the probability, right? So in that case, we get that A is proportional to this E value, because it's essentially equivalent to PMN, right? Or log of PMN. So that's just this right here. And to finish off Beer's law, like, we might ask ourselves, okay, what are the other things that this is dependent on, right? So one thing we know it's dependent on, right, is, okay, this is sort of a microscopic picture, right? So there's one molecule present. So it's definitely dependent on the concentration of molecules in that area. So we have to consider the concentration. So that's definitely going to be part of this proportionality constant. But the other piece of that puzzle is, okay, we also know that there's this thing that exists called scattering, right? So there's a certain amount of light that's going to scatter away, not absorb, and not not just pass straight through the sample, but actually get scattered. And that depends on how long the path length is, right? So those are the two pieces that finish this off, essentially. And we get this epsilon BC, where A is the absorbance, epsilon is the molar absorptivity or extinction coefficient. And again, that has all of these crazy variables in it, right? And it is wavelength dependent. That's a huge key for this lab. B is the path length, right? Which the way our setups in our experiment are done is not going to change. So that is a true constant. It's just the size of the cuvette we're using. C is the concentration of whatever molecule we're measuring is, right? And at a given wavelength, temperature, solvent that we're using, right, this is just a linear relationship between absorbance and concentration. And that's the key. We now have a linear relationship between the absorbance, how many of these photons get absorbed, and the concentration, right? So we can measure the absorbance and infer what the concentration of an unknown solution is, right? So this is done through UV-vis spectrophotometer. This is a double beam instrument, which are the most common types that you see used in research. They're typically more accurate than a single beam instrument like the one you used for a previous lab. Excuse me. But essentially, they have this type of setup. You have a light source that gives you a whole bunch of different light wavelengths, basically. You have a diffraction grating, or a monochromator is another word for it, that gives you only one wavelength that you're interested in studying, at least at a time. Typically, you'll scan a whole bunch of different wavelengths by moving this diffraction grating. But yeah, you get one wavelength, right? Because your molar absorptivity is wavelength dependent. You want to make sure it's the same photon. You have a rotating disc, or a chopper, it's called typically, that is allowing the light to go through the sample cell and a reference cell that just contains your solvent and none of your analyte, and then a sample cell that contains your basically your analyte and your same solvent. So you try to keep the you try to get the concentrations of everything else in solution except for your analyte the same between these two cells, right? And then that goes to another rotating disc that's on the same frequency as this one. So your detector is getting several measurements of the reference cell and sample cell per second, and it subtracts the two. And that gives you basically your difference in absorbance between having the analyte in there and not having the analyte in there, right? And you'll get a spectrum that looks something like this, that's absorbance as a function of wavelength. You can also do kinetics experiments like we did for your previous lab, where this is time and absorbance over time, things like that. But this is how the UV-vis spectrophotometer works. And this is what you would have used if you were doing this in lab for the first part of this experiment. Similarly, for the second part, there's a fluorescence spectroscopy portion of this experiment. Very, very, very similar setup. A little bit more simplified block diagram here than the specifics of the mirrors and everything like that. But you have a light source again. You have a monochromator that comes in and puts out just one wavelength, right? It goes into your sample, right? However, now we're not interested in detecting how much light got absorbed and what's coming through. We're interested in emission, right? So it doesn't make sense to put the detector directly on the other side of this, right? Because we don't care how much light went in. We care about how much is getting absorbed, interconverted. Because remember, if we go way, way back, right, to this Jablonski diagram, if we go way back here, sorry, we have excitation, absorption, right? And then there's some vibrational relaxation that takes place, and then there's fluorescence happening. So these are not the same energy, right? So essentially, again, we get a situation where now, just because we're putting in a photon that's 532 nanometers doesn't mean that the fluorescence coming out is 532, because there's energy loss through vibrational relaxation, right? And that's an important concept that I'll get to in a couple of slides here. But essentially, by going like this, fluorescence can happen in any direction, not just along the beam path, like where we're looking for the difference between these two. So we typically do a 90-degree measurement here, and we look for specifically just a specific wavelength fluorescing. So we have to have another monochromator to get rid of all the other light that's scattering and or fluorescing. Potentially there are other fluorescent transitions as well. And we're detecting just that fluorescent wavelength at the detector, right? Okay, so this is an emissions process. And let me sort of note some of the things that I want you guys to think about as you start to do this. So what you're doing for part one of the experiment is you would create and measure the absorbance of two different dye molecules, red 40 and yellow 5. So you're going to make standards of those. Well, they were done, it was done for you, obviously. And then you would construct a calibration curve for each one of these. Excuse me. Then what you're going to use those calibration curves for is to measure the absorbance spectra of a drink that contains an unknown mixture of both of these. So typically, I think it's like an orange soda or something. But there's certain amounts of red 40 and certain amounts of yellow 5, and you would want to figure out how much of each is in there. There's a couple of things to note as you like do this sort of experiment. So, or as you look at the data, I should say. So Beer's law is additive, and this is in your what's it called, lab procedure packet. They give you this equation essentially, but in the case of non-reactive dilute mixtures, Beer's law is generally additive. So you have your contribution of Beer's law from the red 40 and a contribution from the yellow 5. Right? And those add together to give you a total absorbance. Right? Now, remember, it's wavelength. This is a wavelength dependent quantity, right? B is known for all these. B is always the same. But this E red is wavelength dependent. This E yellow is wavelength dependent. And they're different for each one, right? So you're going to have one set of one calibration curve for a whole bunch of different wavelengths, right? And you have another one for a bunch of different wavelengths. And you're going to have your unknown mixture that gives you a bunch of absorbances, right? And essentially, if you were to pick two of these wavelengths, right, you have a two equation, two unknown situation going on, where you're looking for the concentration of red and concentration of yellow. You know what a mix is from the calibration curves. You know what E red is for one wavelength and another one. You know what E yellow is for one wavelength and another one. So you should be able to solve a two equation, two unknown system to be able to find these values, right? That's, I would say, the most difficult part of this lab is doing that. For part two, there's a different analyte they give you, which is riboflavin, very common in a lot of energy drinks. But similarly, if you were doing this in person, you would create and measure the fluorescence spectra of a bunch of riboflavin standards. And then the second part would be to measure an unknown. So in this case, you don't have a mixture, you just have one thing, but it's a fluorescence spectra instead of a, what's it called? Fluorescence spectra instead of a UV vis absorption spectrum, right? But again, just because this is fluorescence doesn't mean Fermi's golden rule doesn't apply to it, right? Fermi's golden rule, again, if we go back, is just the proportion, or I'm sorry, the probability of a specific transition. So this transition going from here to here, right? It's just the probability of that, right? And that's what we care about, right? How much are we putting in? How many of those actually end up becoming fluorescent photons, right? So we can still have a linear relationship between the two, right? So so in the same way that you did it for part 1, you can do it for part 2. And it should be much easier because now you don't have a mixture. You don't have any other sources interfering. The last thing I would sort of look out for is there's a question at the end of the lab write-up that asks you to calculate the error in your stock solution. And that value was that they give you, they give you like sources of error. Basically, when you're making the stock solution, assuming you did it in lab, right, you had a known concentration, then you had to dilute, so you had volumes you had to transfer, and the dilution in your glassware has a certain error associated with it, which they give you in the lab write-up. So in this case, because this is the concentration of your stock solution, which is a quote-unquote known, right, you're not finding that with the calibration curve, right? So this is not a linear regression type of propagation like you've been doing, right? This is just propagating error the standard way based on an unknown. And there's a description of how to do that in Harris. And any statistics book or online source should be able to tell you how to do that properly, or we could talk about it in lab, but it's a lot easier than actually calculating it through regression. But that's what it's asking for in case you get confused. Because I know there's a lot of different ways that we propagated error throughout the class, and this is a lot simpler than the ways that you've done it. It's probably also the kind of way to propagate error that you'll use the least in actual research. But I believe this lab was originally created for like a Chem 1A course. So it's not quite as advanced level as most of the stuff you've been doing. But anyway, yeah, so I think that's all I really have for you. I'm gonna stick around to answer questions here, and this will be in the recording as well. But in general, if you have other questions as it goes on, please email me. I usually get back to you pretty quick. Although it's been a little worse with my internet not being great here at my house versus at school. But also office hours are still going on, normal Pacific times here. Same Zoom meeting ID you used to get in here. Here's my email. Yeah, but that's all I really have. Are there any questions from anybody? We can open that up to the floor now, I guess. Questions going once, going twice. No? Oh, here we go. Here's some. Sorry. When will lab scores for the past lab? Yeah. Sorry, Anand. Hopefully pretty soon. It's taking me a lot longer to grade than I thought it was. Yeah. But I'm working on it. I'm a 3/4 roughly through them. Yeah, about 3/4 done grading them, but they'll be out as soon as I'm done. Sorry, apologize for that. Yeah. Any other questions? All right, have a great day. All right, I'm gonna stop the recording then.